I'm here at UC Davis Undergraduate Research Conference to look at the poster by Ariana. She was in my BIS 102 class, and now she's in the lab of Greg Lanzaro doing work with Anopheles gambia. Can you explain your work, Ariana?
Yeah, so my project is on the interactions between genetically engineered Anopheles mosquitoes and their vertebrate predators. So just a little bit of background information, malaria is a very harmful disease that impacts millions every single year, and it is carried by the Anopheles mosquito. And so while there have been many strategies to try to eradicate malaria, there hasn't been one that's worked quite yet. However, the University of California Malaria Initiative, or UCMI, has engineered 2 synthetic genes to be inserted into the Anopheles mosquito genome so that they are no longer vectors of malaria. And so when it comes to releasing transgenic mosquitoes into the environment, there's many questions and concerns that come about on how they're going to impact human health, animal health, as well as the environment. And so here we wanted to look at one thing, and that is how these transgenic mosquitoes may impact co-occurring organisms that these mosquitoes interact with and ones that consume them as well. So one main predator of mosquitoes are Gambusia affinis, also known as mosquito fish, and they consume large quantities of mosquito larvae. So here we want to see if these mosquito fish consume these transgenic mosquito larvae, if it's going to impact their fitness in any way. And here we looked at their fitness in terms of growth rate. So to get a growth rate, what we did is we took 60 mosquito fish, and they were between the ages of 24 and 48 hours of age. And every day we would weigh them, measure them, and we'd feed them 100 mosquito larvae, either wild type or transgenic. And from there we were able to calculate a growth rate. And so what we found is despite either having a wild type or transgenic mosquito larvae diet, their growth rate, there was no significant difference in the mean growth rate between these 2 groups. And so this data suggests that despite consuming these transgenic mosquito larvae, it did not impact their growth rate in any significant way. And so while this is good news, mosquito fish are just one of many predators of mosquitoes that interact and consume them. And so future studies should look at these interactions with other co-occurring organisms with these mosquitoes. Now the second goal of the study was to look at the efficacy of the UCMI approach to try to eradicate malaria. In order for this approach to work, the transgenic mosquitoes must be equally as fit as their wild type counterpart so that these beneficial genes can be passed down to future progeny. So one thing to know about these transgenic mosquitoes is that in all larval stages, in the pupae stage, and in the first 24 hours of adulthood, there's a red-eyed phenotype that's observed. And so this raises questions as to their visual acuity. Are they able to see as well as their wild-type counterpart? And this is important because they need to be able to see properly to avoid predators, which in turn affects their fitness. And so here we wanted to compare the predator avoidance capabilities between wild type and transgenic mosquito larvae. And so what we did to do this was we took 30 mosquito fish in individual tanks and we put 50 mosquito larvae in each one, either transgenic or wild type. After 1 hour, we removed the mosquito fish and we counted the number of surviving larvae, the ones that were able to avoid these mosquito fish. And we found no significant difference in the mean number of surviving mosquito larvae. And so this data suggests that despite having this red-eyed phenotype, this did not inhibit the mosquito's ability to avoid these mosquito fish predators. And so it is to note that this experiment was done in standard lab conditions with artificial light. So future studies should consider doing the same exact experiment but with UV light, because in the real world, in the environment, these mosquitoes are subject to UV rays from the sun. And so this may impact their red-eyed phenotype and visual acuity in a different way. So all in all, my project gives us a little more insight into how these transgenic mosquitoes may impact our environment, the co-occurring organisms, and as well as its efficacy. So as we learn more, we'll be able to learn how to use these transgenic mosquitoes as a form of vector control. Thank you. 4 hours of adulthood, that red-eye phenotype is interesting. Yes, it is so trying to learn more about that. Can you imagine what it would have been like before? So I don't know too much about the whole genome, but two things. I think it's like stopworks.org. Yeah, I was working there a lot last winter in the rain. Yeah, so probably that.
I saw a video you posted on Instagram or something. You were talking to the camera. Was that a club or research or something?
Yeah, it's like